The biggest thing to note about pre-mRNA is the 5' cap, which helps bring stability to the pre-mRNA. The spliceosome is made up of other proteins that help splice the pre-mRNA. The sRNPs are made up of sRNA that can help that recognize the splicing sites and also specific nucleotides. There's also something called riboenzyme, and what it does is it acts as an enzyme. It acts as an enzyme that can help splice RNA, that can help splice mRNA. So what happens is once the cutout intron leaves the spliceosome, it's quickly degraded by the cell, and the remaining exons are able to join together. They're able to splice together from the other proteins to form mRNA. So once mRNA is formed, there's a couple of specific modifications that are important to note. First is the 5' cap, and also the poly A tail. These modifications are important because one, the 5' end helps allows for the mRNA to bind to ribosomes, and it also helps protect the mRNA. Also allows for the transportation or the export of mRNA from the nucleus. There's also something specific. There's also something else to know, and this is called alternative RNA splicing. And what alternative RNA splicing is during regular gene regulation, one specific gene can code for multiple proteins. This allows for certain exons to be included and excluded from the mRNA. TRP operon is an example of negative prokaryotic gene regulation. Normally, the TRP operon is turned on. When the TRP operon is turned on, the RNA polymerase is able to attach itself to the promoter. When it attaches itself to the promoter, it's able to aid in the transcription of these regulatory genes. When the regulatory genes are transcribed, it's able to produce polypeptide units. These polypeptide units then aid in the synthesis of tryptophan. Now, when there's an excess level of tryptophan, meaning there's more tryptophan than normal, tryptophan is able to act as a co-repressor. Now, what this means is this co-repressor is able to attach itself to an allosteric protein. Now, this protein is called allosteric because there's two different forms of this protein. There's an inactive form and there's an active form. When this protein becomes activated, it goes through a shape change, therefore making an allosteric protein. So this co-repressor is able to attach itself to this currently inactive repressor. When it attaches itself to the inactive repressor, we see a shape change and it becomes an active repressor. Now this active repressor is able to bind itself to the operator. When the active repressor binds itself to the operator, RNA polymerase is unable to bind itself to the promoter. When it's unable to bind itself to the promoter, transcription ceases to exist. When there's no transcription, we can't create polypeptide units, and when we can't create polypeptide units, there's no synthesis of tryptophan. So this allows tryptophan levels to return back to normal. When tryptophan levels return back to a normal state, this tryptophan is able to detach itself from the repressor. When it detaches itself from the repressor, the active repressor once again returns back to its inactive state. So it goes through a state change again, it becomes inactive. Once again, RNA polymerase is able to attach itself to the promoter. When RNA polymerase attaches itself to the promoter, we see the synthesis of these regulatory genes, which aid in the production of polypeptide units, which then leads to the production or the synthesis of tryptophan, and this cycle repeats. Lac-operon is another example of negative gene regulation in prokaryotic cells. Lac-operon is very similar to TRP operon. However, when we start lac-operon, the operator is turned off. While during the TRP operon, the operon was turned on. When the operator is turned off during for lac-operon, what happens is RNA polymerase is unable to bind itself to the promoter. When RNA polymerase is unable to bind itself to the promoter, transcription cannot occur. When transcription cannot occur, we can't produce mRNA, and when mRNA can't be produced, we can't produce these enzymes. Now, the LAC-I regulatory gene is located located more upstream compared to the other genes, other regulatory genes, and it helps code for this repressor, code for these repressors. Now, when lactose is present in isomer of lactose, allolactose, it is able to bind itself to this active repressor. When allolactase binds itself to the active repressor, when it binds itself to the active repressor, the active repressor undergoes the shape change. And when it undergoes the shape change, it deattaches itself from the operator and becomes an inactive repressor. When it becomes an inactive repressor, the operators turn on, and RNA polymers is able to attach itself to the promoter. When RNA polymers attaches to the promoter, it's able to transcribe these regulatory genes into mRNA. When it transcribes these transcribes these regulatory genes into mRNA, each mRNA is able to code for a specific enzyme. For example, the regulatory gene LAC-Z codes for beta-galactosidase, which helps hydrolyze lactose into glucose and galactose. LAC-Y aids in the production of the permease enzyme, and what the permease enzyme does is it helps transport lactose throughout the cell. Finally, LAC-A aids in the synthesis of trans-acetylase, and currently the function of trans-acetylase in the metabolism of lactose is still unknown. Thank you. The first thing that happens in enhancers is the activator protein binds itself to the distal control element. There's 3 of them, and they form this group called the enhancers. It's also important to note that there's also a promoter, and there's this very vital thing called the Tata box inside the promoter. The Tata box is very important because in the end, it helps aids in the transcription initiation complex, and without the transcription initiation complex, RNA synthesis cannot exist. There's also a gene. Here. So what happens first is the DNA bending protein comes, and it causes the activator proteins in the distal control element to come closer and closer to the promoter, while the general transcription factor and a group of mediator proteins as well as RNA polymers 2 are also nearby. Eventually what happens is the general transcription factors as well as the group of mediator proteins actually attaches itself to these activator proteins, which then, and they also attach to the promoter. When it attaches itself to the promoter, it causes the transcription initiation complex. And when this occurs, RNA polymers is able to attach itself to the promoter, and when it attaches itself to the promoter, it's able to lead to RNA synthesis.